Oh, hi everyone. Hi. Welcome on in. Happy Friday. Chat, there's still a poll, a prediction happening of who the special guest is for today. Chat, happy Friday. We have a special guest on. Who's it going to be? Is it Baby Luna, NMD, Irish Jern, Bit Gaming? Maybe it's Smikes. Maybe it's Andrew. Maybe Thug Shells is coming on, although no one seems to think so. Lita, maybe. Maybe Professor Anduza, Paleontologizing. Guys, get those channel votes in. Happy Buttfest indeed, Smikes. Happy Buttfest. How you doing? Chad, are you ready for the big reveal? Are we ready for the poll is over. We're ready for the big reveal? We're ready for the big reveal. Folks, the correct answer of who is the special guest today with a total of 4,970 channel points on the line. The correct answer is Andrew CZ. Andrew is here today, folks. It's okay, Smikes. It's okay, Smikes. It's okay. It's okay. We're gonna figure this out. We're gonna figure this out, Smikes. We're gonna figure this out. Grimly says hi, Andrew. Yes.
Hey! Riot!
You did threaten to raid us out, Smikes. Okay, so let's go a little bit into just the life cycle of malaria, why malaria is bad and how malaria works. Let us... I promise I did all this sizing earlier, chat. Okay.
Honeydew.
A little bit of a look at malaria. Hi, Yuri. Yuri, that is Andrew.
Mosquitoes are usually vegetarian, preferring to drink nectar, fruit juices, and honeydew.
And we've talked about this in the past of what would happen if we killed all the mosquitoes. Why don't we just go ahead and do that? They pollinate. There's a lot of very important ecological things that they do, and there's thousands of species. So if we wipe them all out, it would be devastating for the environment, not just from the pollination standpoint, but also from the food chain perspective. Fortuitous. No chat. Yes, we'll do a shout out for Andrew. Don't worry, Gwen. Gwen, I got you, Gwen. I got you, Gwen. Oh, that's the wrong Andrew, Gwen. Don't click that one. This is what happens when Andrew doesn't chat in the stream, that I don't know who it is. I did the wrong one. I'm sorry, Andrew. I'm sorry. We'll get you the right one. We'll get you the right one. I'm sorry, Gwen. It's terrible, Gwendolyn Pete. But, so they not only are important parts of the food chain, but they also are important pollinators. So killing all of them would be ecologically destructive. But there are ones that maybe we don't want, which is Anopheles gambiae and Anopheles, or Aedes aegypti. Those are the two that are primarily the disease-carrying vectors.
Only a pregnant mosquito will bite humans, seeking nutrients from blood to nourish her developing eggs.
What's wild about this animation is the way they ended up getting this. There is one gentleman. Thank you, Andrew. He's so handsome and kind. There is one gentleman at Vanderbilt who is able to dissect the entire nervous system out of an intact mosquito with all the neurons interconnected, and they can actually take a 3D image of it and make these kinds of animations. One gentleman, he's in his 80s, retired scientist. Absolutely incredible. How do we know they're important to ecosystems, though? Because they're as a food resource. Now, Smikes, there is the counter argument that there may be some other organism that will fit into that ecological niche that the mosquitoes are playing. And if we wipe them out, nothing would happen. But who wants to be the one to take that risk, right? Because if there's all of a sudden, if something doesn't take its place in terms of food and pollination, you're talking catastrophic effects. Yeah.
Have you gone through The Expanse yet? The sci-fi TV series? Okay.
So we wanted to, but I heard it was stressful.
It's
intense for Lita, and that's why we usually watch the shows together, but we haven't.
It's intense. But one of the background plots was they went to set up a self-contained ecosystem on a floating space station or something like that. And it really only took the smallest imbalance, and everything just started dominoing out, and you know, it looked like it was going to be sustainable.
Don't you like me a little bit?
Ew, I don't even like you a little bit.
Huh, I heard you like me a little bit.
Well, maybe just a little
Handsomeness intensifies. It's because Andrew is here, right, Big Gamey? Guys, go follow Andrew. He's right there. Go follow him on his stream. Go do type trains. Go do all things. So it was like just a little imbalance, right?
And a little imbalance, yeah, and it threw off, and you know, the station had to end up shutting down. You just lose everything, and you don't want to take that chance.
Yeah. Spike says, I mean, the malaria-carrying mosquitoes, how do we know eliminating them won't have drastic effects? So primarily because they're disease vectors in that their primary food source isn't nectar. There are things that eat them, and by the way, hi Leia, like Leia was saying, like bats. But bats also eat other species of mosquito as well, not just the ones that are blood-sucking, but also some of the pollinators. So it might still be taking our risks, Mikes, but at least it would be better than the general feel of we should just wipe out all mosquitoes. And yes, Grimly, they're called that keystone species. But Grimly, the thing is, as soon as you start dissecting more and more into like the depths, you find out that everything is a little bit keystone. Because as soon as you start out wiping out one thing, it always is going to domino. And like maybe the only, even at the top of the food chain, right? Like let's wipe out humans. Then the things that we're using and killing off or whatnot to like keep their populations down, those will start spiking out of control. And you can imagine all of a sudden then they start overeating, and that goes out. It just keeps going and going and going and going. Hi, Zen. Hi, Zintaos. How you doing? Zen, that is the lovely Andrew. Isn't he breathtaking? Isn't he breathtaking? Zintaos. He's going to be here tomorrow as well for a giant barbecue, Zen, with Music Girl, Jimmy, and the lovely NMD96, who's currently at T-Swizzle concert. Speaking of T-Swizzle, folks, someone who will not break your heart is BitGamey. Go drop in a follow. Single greatest father in the world, and also game developer extraordinaire, making a video game called Homeward Hound. Or I'm a dog.
SciAnts know you. You're the cutest, and know that you deserve it, Juno. You a star over here. I'll reverse the card like Uno.
You're breathtaking.
How you doing, Optic Nerve? How you doing? You're crying, Debs. Are these tears of joy or sadness, or maybe a little bit of both? Let's continue, chat. I'm just, again, we're going into the background of there's this new treatment of existing insecticides that mosquitoes that carry malaria have been resistant to. And the researchers did something very easy to them, the drugs, and made them even more potent against animals that were not resistant to it originally, and they also made them really powerful against animals that were resistant. Which, so it took an insecticide that was deemed dead, no longer useful for the purpose of getting rid of these malaria-carrying mosquitoes. They did a very simple. I don't even know what the mindset was to this bit gaming. When you hear about the punchline of what they actually ended up doing, they did this, and all of a sudden, it's even more potent than before, and just killing mosquitoes left, right, and center. And it's easy to do. You can do it in your own home. And it seems to be stable for a long term. And it brings up the question of what other things can we do this to, not just insecticides, but if we have resistant bacteria, or, you know, like in hospitals, can we do the exact same thing to those like antibiotics in the similar manner and maybe make them useful again?
I'm excited to see.
Yeah. It's a hypothesis that they have. The drug has been safety tested in humans, Mike. So currently, what the drug had been used for is mosquito netting in developing countries. So that's unfortunately one of the best ways to make sure you don't get malaria in the dead of night is to put a mosquito net over where you sleep. And they used to lace it with DDT, but DDT has a bunch of secondary effects to the environment, like making the bald eagle eggs really, really thin, right, and like killing birds, killing other animals, and also like there's developmental defects for humans as well. these have been used, Smikes, this drug. Yes, DDT is a carcinogen as well. This one has been approved to be used on the nets, and that it's safe for humans. Now, how safe is it for other animals in the environment? That hasn't really been evaluated. The change in what they're doing to this molecule based on the technique that they're doing hasn't yet been reevaluated, but the hypothesis is that it's just a more potent version of the drug. And that is a little bit, when I was reading about what the FDA recommends, it's a little bit on the borderline of if it needs to be reevaluated or not, because chemically it's the same, it's just structurally different. So we'll see what actually ends up happening. Zin says, can we just invent a repellent that repels the female mosquitoes only? So Tokatao, hello. So Zin, in theory, it should be very easy, right? What you do is that you take the odorants that are attractive to a female mosquito, and it's a couple of things that make them attracted to you. Your body temperature, the clothing color that you wear, the CO2. So like Andrew right now would be extra delicious. He's wearing dark clothing. Mosquitoes love dark clothing. He's also breathing, which means he's releasing CO2, which they're attracted to. He has not imbibed any alcohol, which is a plus for him, because they actually, if you're barbecuing outside and you're having a beer, you're immediately more attracted to mosquitoes. Like, I'm sorry, it's a great afternoon, but they like it.
Does that have anything to do with your blood sugar whatsoever?
So your flavor, like the blood flavor, there is a little bit of preference for blood type, but there hasn't been a correlation between blood sugar. There is like some metabolic off-gassing that you do. So like your skin fundamentally smells different than my skin because of the microbes that we have on it. And there is some metabolism that they have, and they release these volatiles. That doesn't seem necessarily linked to sugars, but rather just the skin population. So that's another thing that mosquitoes are attracted to, is the bacterial population on your skin, which may or may not be, you know, I don't know which one, if they would prefer Andrew or me in this case.
From experience, probably me.
Are you blood type O by chance?
I don't remember.
Okay.
I don't think so.
Yes, so Smikes, their favorite is blood type O. It's the experiments are quite, for lack of a better term, interesting. You give them a Y maze, and you give them different, like 2 different blood types at the end of the 2 tunnels, and the direction that they go in is the one that they prefer. And O is the flavor that they prefer. And it's like, it makes an evolutionary sense because one of the most common blood types. And so it kind of makes sense to evolve in that way. They will, if forced, eat other blood types as well. So they're not above it, but they do have preference for it. Do they enjoy listening to type O negative? So Smikes, music is an interesting question on these animals. There have been some studies on fruit flies showing that oogenesis, or the development of the embryo, is perturbed by different frequencies of sound. So it may or may not serve as repellent or an attractant, depending on like how you're playing the zesty beat, as it were. I never cared to know my blood type. I know it's probably wrong, but I suspect they can get that information from the doctor. Hopefully, Zen, unless you're out in the jungle with Sanji exploring, and you get a cut with from a tiger or something, Zen, and then all of a sudden, Zen, you're rushed to the local village hospital where they have a couple of bags of blood and an IV and no medical records, and then what do you do, Zen?
You hope whoever donated that is a universal donor.
That's right. Otherwise, you're SOL. There's a mosquito right now trying to get my blood. I don't like that, Debs. Be safe, Debs. Be safe. And Yuri, too. My God, y'all are all covered in mosquitoes. But so, Zen, to have a repellent for a female mosquito, you have to get rid of all those factors that we talked about. In addition, body heat is something that they're attracted to. The odor that is released from the microbes on your skin, which you don't actually want to get rid of those microbes. So if you ethanol down your body to get rid of them, it would actually be a very negative health consequence. Heavy breathing, so the CO2 from your breath. If you're working out and running outside, you're actually more attractant all of a sudden to mosquitoes. Drinking ethanol. They're not the only animals that are attracted to ethanol. Fruit flies, other insects. There's developmental and protective benefits to alcohol for them. And then, weirdly enough, mosquitoes are attracted to certain clothing colors. They're repelled by khakis and certain shades of blue, but otherwise, like the dark colors they're quite attracted to, which I was very upset because most of my wardrobe up until I started buying streamer shirts was just black and Godsmack. And
see, it's easier. It's just easier.
It is. That's why I just buy Beans merch, butt fest all the time. Morbid question: will a blood-sucking mosquito 17 bite corpses? That is actually a really good question, Mike. My hypothesis is no. And why is that? If you give a mosquito a tub of blood in its a little box container to try to feed it, it won't eat it. If you give it, you have to have a bunch of different factors for them to eat from any kind of blood source. And so hi Crystal, not just warmth, but you have to put a film over the blood source that they have to have that piercing sensation. So, right, they would do that for like, say, a body, but it's not enough, actually, Crystal, to have a warm source next to it. It's not enough to have it covered with a membrane that they can pierce. It's not enough because you also need a CO2 emitter. So if there is a body, it's not presumably breathing, right? It's not Resident Evil. And if it's not breathing, there's no CO2 released, and then the mosquitoes are not attracted. So, Zen, to answer your question, there is a lab at Vanderbilt. It's the same lab that is able to dissect the nervous system off, or like removed from the body of a mosquito. Their hypothesis is, how can we antagonize the CO2 receptor in the antenna of the female mosquito? So there are antagonists in those antennae that will fire and say, like, this isn't a CO2 rich area. So how do we activate those neurons and therefore override the ones that are detecting CO2? And so they're doing a small molecule chemical screen to figure out, okay, what molecules can we do that act on that neuron, like that act as like a molecule that binds to the receptor in that neuron, turn it on, and then induce the behavior of flying away. Now, Zen, this seems like there's more money and more prospect right now in genetic engineering and modifying the genetics of the mosquito either to be resistant to whatever they're carrying, so that they're not going to carry any kind of bug, right? No pun intended. Or make them sterile. Seems like that's the better avenue, but there are labs testing these kinds of small molecule either inhibitors or activators to repel, in particular, the females. Now, the males, they don't care about CO2. Because they're going for nectar and they're going for flowers, and so you would therefore have that sex-specific, like pushed or one over another. Does that make sense? Then mosquitoes can be jerk. They are extremely cool. Not their fault they cause so much pain to humans. I mean, Gwen, we talked about most species don't cause pain to humans. They're pollinators, right? And so most are never gonna bug you. It's roughly 30 species suck blood, and then the big ones, there's like 4 that carry a bunch of disease vectors. So
If she drinks blood from someone infected with malaria, she too becomes infected with the disease.
Bing says yes, that maybe the decomposing bodies are releasing the CO2. So maybe Smikes, they would be attracted, but the heat body source is still an issue if a mosquito would be going towards it. So as I guess you could, as per Columbo, this is where the murder mysteries come in. If you put like a warm blanket, a heated blanket on top of a body, maybe you could simulate a mosquito coming towards it. I know, exactly. Andrew's got it. It's like Columbo ant.
The tiny drop of blood filling the insect's stomach is teeming with malaria parasites.
This video is 8 years old, and I just want to point out there is something off now about it that we didn't know at the time. This video is saying this one droplet of blood is full of malaria. And these are like little trypanosome creatures. They're small, they're wiggly. It turns out that in the lab, what was done was they were trying to simulate a malaria infection, and they were actually overloading the mosquito with far too much of the pathogen, much more than the animal would ever get in the wild. And so when they were testing effects, it turns out they were not the correct effects. Because if you lower it to a physiologically relevant amount of malaria, the animals completely behave differently. So when we're doing these preference tests, like Zen, if we were asking like, how can you fend off a malaria infected mosquito with this repellent, if you infect it to physiological level versus if you infect it to this high level that they were doing, the behaviors are very different and they will inform you very differently. Which, that's the beauty of science, is that you're always learning, but that's a factor that no one ever thought about was being a thing. So this video is a little outdated on that front. A university in southern United States that does corpse research test decomposition. What is the purpose, Mikes? I'm curious. Hi, Nelf. Welcome in, Nelf. How you doing, Nelf? Nelf, this is Andrew. Please go follow him. He's tagged in the title of the stream today. Click that follow button on him, please. It should be very handsome. He's an amazing streamer extraordinaire. On that partner Poosh as well. Oh no, not the mystery Andrew.
I got it.
Wait, did I put it wrong?
Yeah, I got it. Trying to do too much on the fly. I will say though, the multitasking that goes on.
Together, we can rule the galaxy.
Bean!
Hey, Bean.
Thank you for the resub. Bean, do you see what today is? Hi, Bean. Chad, go follow Celestial Beanart as the Fancy Pants Shadow just says, and go buy her coloring book. And, oh my God, Zedman.
The galaxy.
Hi, Zeddy. Hi, Bean. Thank y'all for the resubs. Bean, 17 months. Zeddy with 23 months. Zedman, 23. 2 divided by 3. 666. The number of the beast.
Bullet the science guy. Bullet the science guy. Science rules. Bullet the science guy. SciAnts is a partner on Twitch. With lovely white Lita. Bullet the science guy.
Thank you, Gwen. Thank you, Gwen. That was made by Celestial Beanart. She does have an artist badge here. It's hidden right now, and she has a little Smikes. For the tier 1 gifts to Ollie Robs, not Oily Robs. Ollie Robs. Thank you, Smikes, for that gift sub. Thank y'all for the resubs as well. Appreciate the heck. I got kicked off a chair. Maybe, possibly. Let's go.
The parasite form that is deadly inside humans cannot survive in the mosquito's stomach and is slowly digested with the rest of her blood meal. However, back in the human host, a few of the parasites turned into a different type of cell.
Hi, Dame Karen. Dame Karen, this isn't just a friend. This is one of the mods, Dame Karen. This is the lovely Andrew CZ, Dame Karen. On top of that, Dame Karen, we are having a mini half mod party tomorrow. Music Girl, Andrew, and NMD96 are gonna be here for a barbecue, and that's like, unfortunately, it's only half the mod team. Couldn't get them all here. No, I'm not cutting them in half, and I'm not barbecuing the mods. I am making a copious amounts of meat, as they should. Are all blood cells the color red, even if they're white blood cells? So the red gives them, it's a function of the iron in them, and the heme group, and it's not that color. Like, white blood cells are actually not red if you put them under the scope. It's usually just when you're bleeding, and it's just the densities are different enough where you can't tell. But if you spread them out enough on a microscope, you will see them. No, Alex. No, the mods will not. No, there's no cannibalism, Alex. Possibly, no. But Barley's been nice to Andrew. I promise, Alex. Maybe. I think. If a parasite cannot survive the mosquito, how does it get into the bloodstream? So Zen, what they were suggesting is, after the mosquito has acquired the parasite that would cause the malarial infection, if the animal is not exposed to a human blood meal, eventually the malaria will be broken down with, like, the parasite will be broken down in the digestive tract of the animal, of the mosquito. Because eventually it will just be pooped out and then it's done, right? But if it feeds on something earlier, because what it splashbacks in is what it is. When it pierces the skin, our cuticle, our skin, the reason you have an allergic reaction is because the mosquito spit in you. And that's what you're allergic to. And so it also spits back some of these parasites, and you don't need a high parasite load for them to start reproducing in your blood. Yeah, I know it's gross, Leia. It's nothing but Gucci. It's anti-Gucci. You can build a giant mod farm to keep them like ants in the yard so you don't have to leave. Just an idea for science. Optic, listen. If I could build a giant like complex where we all had like nice houses, and not because of like mods living with a streamer, but just like our little community, I would do that. But nothing like a farm. We could have a farm on site, but it's not a mod farm. Mods are beautiful people. Modern housing. Oh my god, optic nerve. Hi, Gracie Cat. It is Andrew. He might be being zoomed in here, Gracie Cat, or he might actually be here. We don't know. Hi, Gracie. Hi, Gracie. You recognize the chair? Okay, fine. Fine, Gracie Cat. Maybe we did a green screen and a lot of production value went into today's episode. Or Gracie, it didn't, and he's actually here for a couple of days. He's actually here. Yeah, he is. He is. The name white blood cell derives from the physical appearance of the blood sample enters after centrifugation. White blood cells are found in the buffy coat, a thin, typical white layer of nucleated cells between sedimented red blood cells and blood plasma. Thank you, Alex Vixen. Check out oh yeah, Gracie Cat. Bean and I had our anniversary today as well. 17 months, Gracie Cat. On the day of days
What drink?
of butt festery shirt. But one side you read Smikes' post. When viewed under the microscope, white blood cells typically appear as a clear, pale cells. They lack pigments to give the other cells their characteristic colors. Yes, exactly. Thank you, Smikes. And thank you, Alex, as well. And thank you, Big Gamer, for the hydrate. All right.
One that is sexual, but remains dormant. Malaria sex is triggered when the warm human blood begins to cool inside the insect's stomach. The female form of the parasite matures into an egg. The male form takes a while longer to mature into sperm. This sperm is from an earlier feed.
I like the swimming sounds they had in the video.
It really makes it realistic.
Yeah, of course. It's like, you know, that's what I imagine when I hear a sound of swimming.
The fertilized cell can glide and begins to explore its new environment.
It does, Hugan. It also looks like it a little bit, too.
It migrates to the outer lining of the mosquito's stomach before transforming into a cyst.
Hi, John.
Each cyst produces thousands of thin, tiny parasites which seek out in
malaria and a way to overcome it, in particular with drug resistance. So drug resistance in malaria, carrying mosquitoes is a big deal because you'd have to keep recreating more and more new drugs to kind of combat that. And that's a ton of money. That's a ton of time. And it turns out these researchers, they did something to the drug that made it better at killing mosquitoes than before. And what was crazy, John, is they even killed mosquitoes really well who were resistant to the drug by just doing something so simple that you can actually end up doing it in your own kitchen. That's how simple this is, and which is very important because these are in developing countries that might not have access to like a high-tech fancy lab out in the field. And so if you need to do something very unique and advanced, you wouldn't be able to do it, right? And so I haven't said the tech yet, John. We're getting to it. We're just doing a little bit of highlighting of why this is important, a little bit of background about insecticides in general. We're also going to touch on what other things they're trying to do to fight malaria, and then we'll get on to this particular thing that the research identified. And so again, these cysts.
The next time this mosquito bites a victim, the malaria parasite will ride in with her saliva and infect another human.
Here we go. Does it cover the dormant malaria?
This year, 10% of people on Earth will be struck down with malaria.
What do you mean, Zen? What does what cover? Like this effect happening, or the transmission to the human? Which component, Zen? Hate mosquitoes? Not all of them, Dame Karen. Most of them are pollinators. The former. Zen, I forgot what the former was. I'm so sorry, Zen. Could you write out to which is referring? I lost the train of thought there, Zen.
Most people who die from the disease will be pregnant women and children under the age of 5.
Can we rabbit hole into sickle? We can rabbit hole into sickle after we, I think, finish out the drug component of it. Just because, Smikes, that is a genetic protection that I think is still on topic, but remind me once we finish the drug component of it.
What is our Alzheimer's thing today?
So the Alzheimer's thing today is there is a mutation that if you have, you're gonna get Alzheimer's, and it's gonna be really bad. And there is a second patient that has been identified who has this mutation, doesn't have Alzheimer's, and he's old. And it's like, why does he not have this Alzheimer's? It turns out he has a second mutation that counteracts the effects of the first one. And they're trying to figure out what that effect is and like how, because it's one of the reasons why we have Alzheimer's is that the cells, they build up these platelets or these tau molecules. It's like you have all these like this buildup. And this mutation, 100% of the time, Smikes, if you do a cross section of these brains, like post mortem, there's these deposits all in the brain, and it's considered a hallmark of Alzheimer's. But this second patient, like it's the second one, has this other mutation, and there's no platelets, there's no buildup whatsoever. And so it's trying to figure out what is the molecular reason there's no of this buildup. Oh, we didn't know if the plaque was, so we don't, Smikes. So Smikes, there's some studies where if you get rid of the plaque buildup, there is still the effect of Alzheimer's. And there's other studies where if you get rid of the plaque buildup, it clears the effects. And so it's not a simple, there's something else the mutation's doing. It is not at the stage, the research isn't at the stage where they actually put it into a mouse, because that's what would be the next step, is because you can't do the genetic testing on a human. You'd have to look at what the molecular mechanisms are in the mouse. Unfortunately, when you put in some of these human mutations, it doesn't necessarily mean it's going to simulate the effect of what you're trying to study. Sometimes there's very different effects as well. Alex, have a good night, Alex. Thank you for hanging with us. So that's the Alzheimer's story tonight, and it's really cool. It's still a lot left to be desired in terms of like identification of what's going on, but it's a potential genetic trick to overcoming the effect of Alzheimer's. It's a hope. It's you know, it hits home for us because of Lita's mom, and from her diagnosis, we could tell she doesn't have this second mutation that would be protective, but maybe there would be, if not for her, someone else that could be treated. Because the idea is, if you can simulate a drug that does what the genetics do, then maybe you can treat it as well.
Yep.
Or gene therapy, but that is so hairy because you have to hit all the cells that are being affected, and they all have to integrate into the genome, and you have to make sure that when you're putting in this genetic piece of information in the genome, you're not disrupting anything else, inducing more mutation. It just snowballs. So like that's why drugs are easier.
Yeah.
And also not, because you can't
target.
Yeah.
Yeah.
Or unless you wanted to have like a brain tube that you're constantly injecting, and that you can't live like that. So there's a lot of factors that go into the drug discovery. Are watching the parasite being formed? Yes, in the mosquito, Leia. So the parasite is acquired by the mosquito from its food sources or when by infecting another human. Or sometimes it also lands on poop, and the poop is a way that the parasite's acquired. It is not in its mature state. It's just the eggs that are ingested first by the mosquito. That egg hatches, the organisms mate inside the mosquito, and then you have these adult trypanosome-like creatures that are then deposited into the next victim, as it were, for lack of a better term. Why doesn't the parasite kill the carrier? Is it symbiotic? It has no negative effect on... It's not designed to affect that host. It's that host is an intermediary, and that's why it doesn't kill it. There's other animals like schistomiasis. I don't remember how to pronounce it. It's the carrier is snails. The exact same idea is that the snails are the carriers, and then they're eaten by birds or something else, and then that's where it kills the host. And so it's just there's a reason for the carrier. It's not malaria snails. It's schistosomiasis, I think is how you say it. It's another like terrible disease state. Yes, astronomy show. You're asking about Buttfest. Buttfest is put on by Celestial Bean Art, with our amazing friendos. And by the way, guys, go check out astronomy show as well. She had this amazing festival where it was a bunch of artwork that was featured. She had a special emote for it. She had an interactive carnival-like atmosphere. She made a digital carnival where you could play games live on her stream as she had a VTuber model. She even had an interactive bathroom where you can draw on the bathroom wall. It was an there were guest stars, astronomy show. Thug Shells performed. I think Pineapple Hoops was there. Our Avatar The Last Lightbender was there. There were a lot of people there on the... I even gave butt facts. Yeah, about butt goo. And it was a 2-day event. Professor Knight was there. It was an absolute riot of a time. It was beautiful.
Speaking of Professor Knight, that's one of his emotes.
It's brand new.
We here living our dreams. Chemistry, cause we rocking out with science dreams.
That's a beater.
Science, but we ain't bugging. We thugging. I know that we up here making sure that the beat is snug.
Bean said, a fun fact, I would submit an application to a Buttfest live at TwitchCon. Bean, what does that mean? Like, is that like you submit it for a panel, or like an event like the Performance Thuggy had? Like what kind of submit? Because that sounds awesome.
Yeah, it does. Yeah.
And you'd be highlighting creators in the community, which I would assume they'd be all about, because it's just like a bigger form of having that like side stage.
And that has some stopping power too. You go, what's Buttfest?
Yeah. Oh yeah. The name immediately grabs it. Apply for a workshop, do a demo, and then build it. Bean. Bean. If I had any pull, man, I would contact people. I have no pull, Bean. But that sounds incredible. That sounds absolutely amazing. And Bean, I would wear the Buttfest shirt. Or Bean, actually, I would have you make a special SciAnts at Buttfest shirt, because I know we don't like to talk about this. I still need to advertise SciAnts, but then that way we can have a SciAnts Buttfest or something like that. So it's like crossing the streams, literally. It's all kinds of Gucci. I gotta... A beanie. Listen, Dame Karen, on Celestial Bean Art's store, there is a beanie with this design as well. It's a beautiful yellow beanie that Thug Shells was wearing as well during some of her live streams. No, you can.
You are wholesome as ferret. Yay!
Bean, were there any archives of the Buttfest stream? Would you, Bean, if you have it, I don't know if it's on your YouTube, Bean, or somewhere, if you wouldn't mind linking it for Astronomy Show, so they will be able to experience Buttfest. Astronomy Show, I think the easiest thing for you to do, though, is just go follow Celestial Bean Art and have her recount it, the Buttfest, because she does it much better than I could ever do it. I was just hanging out, enjoying myself, you know, but like all the behind the scenes things and like, you know, it's just follow Bean, just follow Bean. It was epic, Smikes. It was epic. I even, like I said, butt goo. We talked about that. There's a lot of butt science as well. The whole streams are highlighted. I can give you the time. There we go. Astronomy Show. Follow Bean, and they are highlighted and they're ready to go there. Thank you, Astronomy Show, for following Celestial Bean Art. She's also on her partner Poosh, so make sure whenever she's live to go lurk on her. Say hi, go into lurk mode. It's totally Gucci, but let's support our best Bean and push her to that purple check mark. But they should be Smikes, but they're usually put down and not celebrated. It's very sad. It's very, very sad. But you know, one day we'll get over it. Resident Butt. I prefer buttologist, Bean. So,
the malaria parasite is...
Let me just double check. Yes, so now you might be wondering how we go from the malaria within the mosquito to the human. And so now we're going to go ahead and do that component, and then we'll talk about, you know, why, like how to counteract it, what to do about it, and then we'll get into the actual...
It's an ancient organism. It has been with us since before we were human. Famous victims include Alexander the Great, Genghis Khan, and George Washington.
So for you, Linux, which you coming on in, we're talking about malaria because there's a new study that's looking at different ways to fight off malarial carrying mosquitoes with existing insecticides that used to be resistant. How do we know Genghis Khan had malaria? Journals and diaries? I believe so. Same with Alexander the Great too. I know for him it was these the journals and the description of like fever and the discoloration of his body and like when he died and like whereabouts they were exploring. Not so much in any other terms of like really like there was no like visuals of it. But it was really well written down, and it's, you know, it could have been something different, but given the area that they were located in, the common deaths that were occurring as a function of it, it's most likely it was malaria. Guys, also go check out the lovely and amazing, as always, Supernatural Writer. Go check out Super. Gotta have an anti-malarial for a genetic disorder. Linux, hopefully this will help with the malarial infections as well. Let's see, hold on. Smike says, transfer from human to human through orifices. You'd have to have blood-to-blood contact, Smikes, and the malarial load is not so dense that you will necessarily be guaranteed of it. At certain amounts, maybe in certain like densities within your blood, but otherwise it's not as high level of density as, let's say, AIDS would be, where it's much more likely to have it. It's just the viral load is different because you're having a parasite versus a virus, and it's different reproducing. After
the malaria life cycle follows a devious path.
After the mosquito lands, transmitting the malarial infection to a human host. My dad goes commando. He's quite vocal about this fact. That's okay, Leia. That's okay. I just can't. I just personally cannot. It's a mental block, you know. Being a swimmer for so many years, you don't go commando in the pool. Yeah. I know, Bean. It's, as Lance says, to the sound is also a little bit, you know.
Swapping back and forth between mosquitoes and humans.
I will say that their anatomy is really cool. They have, and we've found this underneath the amber, right? They have the long proboscis, which they use to suck up the blood, and there's these two little, like you can see it in the animation. These little almost holders. They like they support the
I'll be
structure.
back.
Thank you, Miss Game. They support the structure for the animal to drink up the blood, or not the hemolymph, the blood. The hemolymph is the insect equivalent. And it's just cool that they have this guiding force to it. And the proboscis is also a sheath for the actual puncture source. So it's like, it's a very complicated system. It's very irritating, but it's a very beautiful and elegantly built system. And that's some of the things that we look for in the amber is, are these additional features there? If not, if you just as a proboscis, then we know it's not a mosquito. And you might be like, well, Belen, what if it's just a more ancient mosquito? That is very possible, but not 30 million years ago. Maybe even further, like 90, 100 million plus. Maybe there were ones that didn't have this feature, but 30 million, it had been very much commonly like settled in.
This mosquito is infected with the malaria parasite. Because she is pregnant, she has become hungry for human blood. During the bite, she injects saliva to stop the blood from clotting. Her infected saliva also carries the malaria parasite. The parasite rides the bloodstream like a network of roads.
So if y'all have noticed that after you get bitten by a mosquito, I'm assuming most people here get itches, assuming you do too. If you notice the size, like what causes the different size bites, that is how long the mosquito took to find a blood vessel. So if it's a bigger size welt, then it's because it was searching around more and more, and there's more mosquito saliva that has been injected into you. If it's extra gross, right? So if you got a big welt, that one spat a lot into you. And so they search around because there's sometimes blood vessels not close by, and so they have to search and they're probing. And so right here you can actually see this strand here, that's not a blood vessel coming out. This is like the unsheathed portion of the proboscis, and it's probing underneath your skin trying to find a blood vessel. And yes, Smikes, we are allergic to their saliva. That is why you get the welt. If you do this to a mouse, that's a common thing in the lab. You can shave the mouse belly, put it to sleep, and they will have their blood sucked, and they wake up, they have no allergic reaction, they don't even know anything happened, which is cool. And unfortunately we can't do it on us. In the wild, Dame Karen, they have a preference for humans. They don't have to feast on humans in the lab. You can give them cow blood, you can give them mice, and then there are labs that, Dame Karen, it is mandatory to put your body part in there, either your arm or your leg, and feed him that way. Now, Dame Karen, to me, that seemed like a little bit of a excess, like something I would like to do, right? Because it's, I don't know, I don't like getting eaten by mosquitoes, but at the time, I was told I asked them, like, some of the lab members, like, do you have to do this to join this lab? And they're like, well, it's not on paper, but you have to do that. And then, Dame Karen, my questions were to the professor. I'm like, is there any effect of blood type on your experiments? Because they were doing different experiments. And if you don't have consistent blood type and you're doing RNA analysis or behavioral stuff, and one's an AB and one's an O, could that have an effect? She's like, I don't think it has an effect. To me, that's not sufficient enough of an answer. Like, I'd actually want to test that. And that's why I think doing, like, either the mice, which you can get genetically identical mouse lines. And those mice, they won't get the malaria. They're not the natural hosts of these animals. Like, it's not that their immune system fights off the parasites. The parasites just don't have the right ratio of, like, their metabolic needs are not met inside of a mouse, and it's just like a host incompatibility issue, not a great immune system fighting it off. And so yeah, like mice are genetically identical. You can keep ordering them; they'll get the exact same line. Same thing with the cow blood you can get from a company, and you can get the exact like this kind of cow line that you're getting in there. Apparently they don't like it as much, but you can do it. Other primates, I do not know, Smikes. I wanna. My gut says no, that there is still incompatibility between them, but it wouldn't surprise me if certain species of primate that are close enough genetically would still be hit. But my gut says no.
Seeking its first target. The core of your body's blood filter system. The liver. Sensing its arrival at the liver, the parasite searches for an exit. A sentinel Kupfer cell is the entry point to liver tissue.
Yeah, they don't have the same noise as sperm, is it?
Leaving
No,
the
I mean,
blood,
it's different
the
organisms.
parasite
You'd expect it to swim differently.
Yeah.
infects a liver cell, killing one or more other cells on its way. Over the next few days, the parasite undergoes hundreds of nuclear divisions, copying its DNA over and over again. A single infected liver cell can create thousands of new parasites. The next generation of parasites are modified to infect a new target. Red blood cells.
And this part isn't really fully understood either. It's hypothesized to be epigenetics. The genetic code for seeking out red blood cells or liver is in both of these animals, but in one it's turned on for one, and then it's turned on, the other one is turned on in the other, which is, there's some switch there, and how that switch works, and like that's still poorly understood, because it's, you'd have to look at it in a human model, and you can't ethically be infecting humans. Hi, Humbler John. Guys, go check out the lovely and amazing maker and crafter extraordinaire that is Humbler John, who's also going to be a 3D printing wizard. How you doing, Humbler John? They have not mentioned the species in this one, Gwen, but it is an Anopheles gambi. That's primarily one that transmits malaria. In the paper that we're talking about, they looked at a North American Anopheles, and it's an Anopheles quadrimaculatus. And why did they test that one? It was primarily because that one had resistance to this drug. And that's why I tested it. The clear is tricky. Humbler John, if anyone could do it, it's you. Because you're humble, and lure, and also John. I love you for it. Yeah, I did. Leia pointed at the size of the body. For the West Nile, so that is a different flavor of mosquito, Leia, that carries that one. The idea, though, for this this approach that they did with the drug would be applicable to any. This drug doesn't act on this just one species of mosquito. It's more of a universal effect on mosquitoes. So in theory, it could be used for ones that also carry West Nile. Thank you, Humbler John. It's my Bloodfest shirt. Isn't it neat? I think Schmakey's was actually wearing one recently too. Yes. Yeah. So and then he and I wore our Thuggy shirts the same day too. Different shade of blue, so we weren't totally twinning.
It was perfect. It was perfect.
Trying hard.
Absolutely perfect.
No.
Inside a red blood cell, the parasite can hide from the body's immune system. The parasite slowly devours the contents of the infected cell and creates more parasites. The infected cell becomes sticky and grips onto blood vessel walls. Once mature, the infected cell bursts, spreading more parasites through the bloodstream. Malaria victims suffer fever, loss of blood, convulsions, brain damage, and coma. This year, 10% of people on Earth will be struck down with malaria. Countless millions have been killed by it. Most people who die from the disease are pregnant women and children under the age of 5.
And that's in part due to immunity and then the stress that you have on your body when you're pregnant, about the amount of blood that's being also moved around. So, we all can agree, this is the life cycle of malaria. Malaria is bad. It kills people. What are folks, researchers trying to do now? There's two approaches. One, insecticide, which is the big thing this paper talked about. The other is genetic engineering. So we're going to look at the genetic engineering side of things first, and then we'll look at the drug side, other side of things. It's the two different sides of fighting off this terrible infection. And I know we're going about it a long way, folks, but I hope it frames the argument of why this kind of study is important, why serendipity is important as well. I think there is a quote here as well of why they did this. See the quote. Yeah, so the researchers didn't set out to revive the insecticides. So that's what they actually ended up doing. They were working on crystal growth experiments because the drug, it's a solid and there's crystals underneath the microscope. When you look at it, you can isolate also a crystal structure from them, which is you can do with any solid. And they were trying to look at the crystal structure of the drug and see how it interacts with its potential target. And they just so happened to find a way to make it better, which is, we'll jump into that in a moment. But let's see what the geneticists are doing. Kill the mosquitoes, I don't care if they're pollinators. Dame Karen, the world will implode.
What if you could use genetic engineering to stop humanity's most dangerous predator? The deadliest animal on the planet, responsible for the death of billions, the mighty mosquito. Along with other diseases, it plays host to malaria, one of the cruelest parasites on Earth, possibly the single biggest killer of humans in history. In 2015 alone, hundreds of millions were infected and almost 500,000 people died. A new technology could help us eradicate malaria forever. But to do so, we need to engineer a whole animal population. This is not a hypothetical problem. The modified mosquitoes already exist in a lab. Should we use the technology? And is malaria bad enough to risk it?
So the reason that the numbers are different, Smikes, one is infected and the other is death rate. And so those are the reasons why there's those two different numbers. The times and ages of the video are pretty similar. So I don't think it's that they were necessarily looking like it hadn't changed that much. I think it was, again, infection rate versus actual death rate. Dengue, West Nile, Zika, right? So there's a whole bunch of vectors that they're carrying.
Malaria.
The death rate per infection, I'd imagine we could do the math, Mikes. But it's all estimates because there's probably a bunch of people that are also getting infected that we don't account for. And so I don't know how good the data is for that. It's like what level of, how accurate are the models? Without Lita, we could do the math. I don't know. That's hard, Smikes. Not without Lita. She's very good at the math side of things.
Malaria is caused by a group of microorganisms, Plasmodia. Very weird microorganisms that consist of just a single cell. They're parasites that completely rely on mosquitoes. Malaria always starts with an insect bite. In its salivary glands, thousands of sporozoites wait until the insect penetrates your skin. Immediately after invading you, they head for the liver, where they quietly enter big cells and hide from the immune system. For up to a month, they stay here in stealth mode, consuming the cells alive and changing into their next form, small, drop-like merozoites. They multiply, generating thousands of themselves, and then burst out of the cells. So thousands of parasites head into the bloodstream to look for their next victims, red blood cells. To stay unnoticed, they wrap themselves in the membranes of the cells they killed.
Which is, I love the animation style. It really brings across the point. According to the World Health Organization, estimated 241 million cases of malaria worldwide, resulting in 627,000 deaths. Fatality rate 0.26 globally. However, it's important to note the rate can vary significantly depending on the region's specific circumstances. And I don't know how good the estimate is for the infection rate, Smikes. That's what makes me wonder whether or not that's how good those models are, especially depending in different parts of the world. And that's a more recent one. These are older spikes, so I wonder if their preventative measures were not as good at that point versus like how good they are now, like the nets and the other preventative metrics that we have in place. I mean, just because the WHO, I don't inherently buy that argument, because we have to, I think, regardless of the name recognition, we have to keep them accountable. And I still want to look and see how the data was made. Here at night, we have trucks with flashing lights. Shoot clouds of mosquito poison at night. Makes me go by the house when they go by. I wonder if it's effective. Astronomy show for us, they do, as they're driving, they're spraying like the bodies of water as well. To kind of wipe them out. To help with that. No, not your bad ever, Smikes. Don't you dare say that, sir. Don't you dare say that, because I'll be very upset with you. Hi, Kibbo, or Kipo. There's Andrew. Welcome in, Kibbo. Talking about some malaria tonight.
Imagine that. Killing someone from the inside and then taking their skin as camouflage. Brutal. They now violently attack red blood cells, multiplying inside them until they burst, then finding more red blood cells, and this cycle repeats over and over. Pieces of dead cells spread lots of toxic waste material, which activates a powerful immune response, causing flu-like symptoms. Among the symptoms are high fever, sweats and chills, convulsions, headaches, and sometimes vomiting and diarrhea. If malaria breaches the blood-brain barrier, it can cause coma, neurological damage, or death. The parasites are ready for evacuation now. When another mosquito bites the infected human, they get a ride. The cycle can start over. In 2015, the Zika virus, which causes horrible birth defects if it infects pregnant women, spread rapidly into new areas around the globe. It too is carried by a mosquito.
I need some science. I need to stop reading comments and science vids on Facebook, Instagram. Yes, Kennedy, do not read the comments on Facebook or Instagram of science videos. There will be some interesting things there for sure, and there will be things that make you want to comment back. And at the end of the day, it's not worth doing. I had one of those many years ago, and I stopped because there was no point to doing it. How you doing, Kennedy? Welcome in.
Mosquito is the perfect carrier for human diseases. They've been around for at least 200 million years. There are trillions of them, and a single one can lay up to 300 eggs at a time. They are practically impossible to eradicate, and the perfect parasite
is this really cool. So it's Science News of the Week. Thank you, Neuralicia. I'm actually going to keep this. Sweet. I'm going to keep this because I'm curious about that as well.
It's science.
It's a little bit of science astronomy show. So this paper, it's open access. How dare in Nature? You know what that means, Neuralicia? It means the authors spent $12,000 to make it open access. Yeah, $12,000 of your taxpayer money to make it open access.
That's got to be grant money. That's not their personal money.
It's not their personal money. Yeah, because Nature had this whole thing a year or two ago that they were making an option for immediate open access, and it was like they were this pioneer of like allowing it to be, and yeah, it's $12K. Leia, no paywall is great, but there's a lot of labs that cannot afford to publish now in Nature. So they imagine you get an NSF grant that's $200,000 for 4 years. That sounds like a lot of money, but if you hire a postdoc, that's $50K a year. That's with their benefits. For 4 years, you haven't even paid a dime on an experiment, right? So let's say you can't afford postdocs. So you afford a grad student, let's say it's $23,000 a year. Let's say you get them to TA, so it makes them $15,000 a year, and then you could do those experiments, and then all of a sudden you do one big experiment, and then even if it's big enough to publish in Nature, you'll never be able to do that. So, so the postdoc's not making 50K smikes, but with their benefits and everything, that adds up to that rough amount, right? Because like there's health insurance and all that jazz. So Leia, like, it's great to be open access, but when you're essentially gatekeeping the existing scientists and making the rich get richer, because the only way to get a good grant is through having a big paper, Cell Science or Nature. The only way to get a good paper is to have a lot of money, and so it's just a circular argument, unfortunately. Looks like they are the UK. I'm okay with them spending the British taxpayer money. Fair enough, Dr. Nero. But the same argument could be put forth for them as well. Right. Nature's overcharging. Dame Karen, there are journals that also charge for color figures. And it's because historically they were in print. And print cost more money if it was in color. Sure. They're no longer in print. They're only online, and there's still that charge. And you're talking $100 for black and white and $500 for color. And that's per figure. I remember we had a paper once, we had to change all the figures to grayscale, and they looked awful, but it was such a big price difference that you're just like, you just can't afford it. But this paper for the Raiders coming on in, we're chatting about insecticide resistance in mosquitoes that carry malaria, and there was a revitalized way of utilizing existing insecticides that the animals were resistant to, where using certain concentrations of insecticide didn't kill the mosquitoes. They did something to the insecticide, which we're getting to, that actually revitalized, like changed their chemical property, and all of a sudden they became even more efficient and efficacious against the mosquitoes carrying the malaria. So we've been talking a little bit about malaria. We've been talking about now the genetic tools that we can do to counter malaria and mosquitoes, and then we're going to get to the insecticide front as well. Per page, no, Leia, not per page, per figure. And a figure isn't full page. So article figures, here we go. So like this right here. See, it's just the bottom of the page. It would be a flat rate of $500, let's say, for a particular paper. Again, some journals had a different amounts, but one that we published and still had this historical front to it. And then there was a per page charge of just print, but that was away from the figure count.
But today we have new revolutionary technology that could enable us to finally win the war against them. CRISPR. For the first time in human history, we have the tools to make fast, large-scale changes to entire species, changing their genetic information as we please. So, instead of attacking isolated groups of insects, why not just change the types that transmit diseases? Using genetic engineering, scientists successfully created a strain of mosquitoes that are immune to the malaria parasite by adding a new antibody gene that specifically targets Plasmodium. These mosquitoes will never spread malaria. But just changing genetic information is not enough. The edits would only be inherited by half the offspring because most genes have 2 versions inside the genome as a failsafe. So after just 2 generations, at most only half of the offspring would carry the engineered gene. In a population of billions of mosquitoes, they would hardly make a difference. A genetic engineering method called the gene drive solves this problem. It forces the new gene to become dominant in the following generations, overpowering the old gene almost completely. Thanks to this twist, 99.5% of all the engineered mosquitoes' offspring will carry the anti-malaria edit.
And the gene drive isn't something that was invented in the lab. That is something that exists in nature. They just connected it to the gene that they're putting in. So it's a way that you can hijack their existing biology to put something in that you want to be there. Yep, we can do to all of them, Dame Karen. So there's there's a small percentage that the gene drive doesn't hit, but if this much of the next generation is now immune against malaria, eventually after a couple of generations you're wiping out more and more of the ones that are not immune. The question then is, as Kennedy was like, what could go wrong? Is if all of a sudden there's some immunity built up, like resistant, like if the malaria evolves now, right? Now the malaria now has the resistance against the immune competent ones, and then you're just like continuing on. So you constantly have to keep putting in more and more members of the population to keep, you know, the malaria from evolving faster than what you're being able to put in. It's called the Red Queen hypothesis, where essentially both the pathogen and the infected are running in the same space, and nothing's getting ahead of one another, and just keeping the status quo. Hi, Spectreflight. How are you doing? Hello there, General Kenobi. Hi, welcome in. Like in Resident Evil? I don't know what you mean, Spikes.
Hello there.
Hi, Spectre.
Hello there. Hello there.
Spectre, this is Andrew. Do you know Andrew CZ Spectre? Was that the Red Queen? Alice in Wonderland? Assume? Assuming it changes before the moment, it could save many lives. It could, Dame Karen, yes. Guys, go check out Andrew CZ. Go. Red Queen is computer AI and Resident Evil. Ah, so it is very different than Spectre. Thank you, Spectre.
If we were to release enough engineered mosquitoes into the wild to mate with normal mosquitoes, the malaria-blocking genes would spread extremely quickly. As the new gene becomes a permanent feature of the mosquito population, Plasmodium would lose its home base. Scientists hoped that the change would be so fast that they could not adapt to it quickly enough. Malaria could virtually disappear. If you take into account that nearly 500,000 children are killed by it every year, about 5 have died since this video started. Some scientists argue that we should use the technology sooner rather than later.
Like you were saying, Dame Karen, there's actually... They did a controlled release of these animals in Florida. And it was on a very small location to try to see what the population dynamics are going to be. Like a very small release. If there's anything negative that happens, it's contained. And actually the biggest issue was public perception. That a lot of people were against it because you're releasing genetically modified organisms. Like, and yes, I know that there's the argument that everything's genetically modified because you're breeding for certain traits, but because this was made in a lab, their PR department was not good enough to overcome that initial hurdle of trust from the general population. And so you need public buy-in to release something like this. And so, Dame Karen, you need to do it on controlled sites, and the countries where this is a big problem. No, we can't just say Florida is Florida. Those are people too, and they represent what many populations are going to feel. There is a huge distrust in science because of how certain scientists talk to different groups of people. That's like that meaning of like, we're talking down to you because you don't understand anyway. And that mentality isn't just in Florida, it's in a lot of places. And you have to be able to explain this. You cannot just start releasing vectors that you've genetically modified without public buy-in. And if you're releasing it with mothers and babies, that's called coercion.
Yeah.
You cannot just tell them this is going to save you because there is no guarantee that it is.
Yeah, I mean, we're still, we went over unintended consequences like before. We know that that's a thing. So if you're trying to get the buy-in of the people, it's not a transactional thing, right? It's not something that you get to force on someone or you know, it's not coercive. It should never be coercive, right? The reason you're getting the buy-in of the people is because you want to engender that goodwill, right? You want to make sure that these people are receptive to this so that you can have successful experiments with this, right? Because if you have people knocking down your doors, you're not gonna be focused on what is the science here, is this helping, is this hurting, is there something we're missing. You're gonna be slammed with PR issues, and you're gonna be slammed with media and slammed with complaining mothers and concerned mothers, right? So that the safety and the concern of the people who are affected is always going to be the number one thing to address.
And as Kennedy points out, there have been science done by organizations and governments. Historically, they're very questionable. Like, people in Puerto Rico were experimented on.
Yeah.
And so, having a government tell you that we're doing this and we're releasing this for your own good, even if people are dying, like you don't want to just you don't want it just to be jumped, right? Because you want it to be like, you want the buy-in of the people for that. Spike's right. There are legitimate concerns, and then there's like long-term ramifications as well, and then there are also the unfounded side of things, the fear-mongering side of things, and both have to be taken into account. And so you can't just loosey-goosey release it. And I know, Dame Karen, if it's a way to save lives, there is the argument that we should just do it anyway because we're going to save a life. But as soon as you bring in people with power, there is no monetary gain in just saving lives. And as soon as that starts coming in, there's going to be changes that are going to be, and you have to be, it's just better to bring in the buy-in of everyone.
Well, if saving lives is your bottom line, we get rid of cars today, right? But that's never going to happen because you don't have a PR team that's good enough to do that.
And as Astronomy Show and Smike's saying, testing on a small scale first, because I mean, Smike's, I agree with this is like the extreme, like if we release this mosquito and 1 billion people die.
Yeah.
I don't think it's going to happen. But if all of a sudden there's some kind of outbreak,
yeah,
or it wipes out the wrong mosquito population for something it shouldn't, but that'll be a big, like an even worse thing, right? And if that happens, there is no recovery from that, from a PR perspective, that we released something that was supposed to help and it wiped out a population. And I know this is one where it's coming to be a little bit circular argument of like, well, but like the data says this, but you still have to go through and very cautiously test everything before you can justify releasing it. And I know part of it shouldn't count as like, why are we taking into account so much of the PR? But there have been a lot of experiments and potentially life-saving things that have been slowed down, if not halted, because of how the PR was treated. And it's like, if you don't get public buy-in, it's not going to happen. And so it's a very important side of the science that needs to be addressed, which is also why if scientists were just better science communicators, we probably wouldn't be in a situation where we have to think so much about this. And also, but there's also a lot of historical things too, Dame Karen, where there has been good reasons to not trust the people in power because of what they did to the groups of people that were taken advantage of. If cars were eliminated, getting people places safely would suffer more deaths. I think Kennedy, Andrew means like from a lung health perspective, from the environment perspective, but you're right, like rescuing people from natural disaster you need transportation for, but your day-to-day lives, there's been a lot shown to be like negative health consequences on that front. But again, no one's going to buy into it.
No.
Because we can't even stop, like, I drive around, I'm not going to stop buying gas or get an electric car, not because I don't want one, but like price prohibitive and, all those factors.
The mosquitoes themselves would probably only profit from this. They don't have anything to gain from carrying parasites. And this might only be the first step. Malaria might just be the beginning. Different mosquitoes also carry dengue fever and Zika. Ticks transmit Lyme disease. Flies transmit sleeping sickness. Fleas transmit the plague.
So imagine if you were able to use this principle to cure all these vectors and like the insects of these diseases, and you can wipe out then not just malaria, but you'd be able to wipe out a bunch of different one of these factors, right? So Dame Karen, while I 100% agree with you, it would be a great idea. Do it on a small scale. If it works, you can apply it to a much broader range of animals that are also employing this very similar methodology of like as a carrier. The mosquitoes genetically engineered to be less harmful to people. The alternative, Dame Karen, is they are killing them by way of sterilizing them. So some of the genes that they're adding into lead to sterile males, and they still try to mate. It's just the females don't know that they didn't get successfully inseminated, and that is something that they're also pursuing. So there's at least two avenues in terms of the genetics that they're going for. One is this: can you cure the animal? The other is make them infertile. Like on one side, either usually the male side of things gets easier to pass on that gene, and then lowering the population artificially. Probably the best thing would be to do both. Right. Mass spraying DDT that saved everyone from mosquitoes, and that worked out great, but not so much right now.
We could save millions of lives and prevent suffering on an unbelievable scale. So, why haven't we done this yet? For one, CRISPR editing is barely 4 years old, so until very recently, we just couldn't do it as fast and easily. And there are valid concerns. Never before have humans consciously changed the genetic code of a free living organism on this scale. Once we do it, there is no going back. So it has to be done right, because there could be unwanted consequences if we set out to edit nature. In the specific case of malaria, though, the risk might be acceptable since the genetic modification doesn't make a big change in the overall genome, it only changes a very specific part.
But as they pointed out, unintended consequences, even a small change, could have big consequences.
I do like that the cuteness was unharmed, though. For the graphic, the cuteness is still there.
I mean, yeah, absolutely, right? Mm-hmm. Without it, I guess
that was the important part of the PR campaign that they forgot to mention.
You know, the good thing about these like this group, is that they do make really good animations, and it makes it easy to process and understand. Because without, like, imagine trying to explain this with like overly accurate, like. Yeah. You don't know what's going on there.
We've watched some of those videos on your stream before.
And it's hard to dissect, right? And what it means.
Very difficult.
Yeah. I'm sorry, Dame Karen. We don't mean to hurt your heart. It's just the cautionary tale that goes into a lot of these things.
The worst case scenario here is probably that it might not work, or that the parasite adapts in a negative way. There is still much debate. Technology as powerful as gene drive needs to be handled with a lot of care. But at some point, we have to ask ourselves, is it unethical to not use this technology when every day 1,000 children die? Humanity has to decide how to act on this in the next few years. The public discussion is way behind the technology in this case. What do you think? This video...
I do like the birds with a DNA sword as well to start off. So I think for the what do you think, well, we have a lot of different opinions in the chat, and I don't think there's one right one. I think it's probably a merging of all of them, really. It's if you had better explanation ahead of time, then maybe we'd be at a position where we'd be more readily do it faster as well. A lot of malaria in the DC area for a long time. Clean it up and take... So the third option, Smikes, is... on the scale that we would need it to, I don't think it would be possible, because what would end up having to do is it's standing water, right? So there are approaches of trying to get rid of standing water in like more island nations, and breeding grounds of mosquitoes, and really wiping it out on that front. The issue is in habitats and climates where water is considered as really valuable, and they leave out standing water. Just because if there's not active plumbing, right? Like, how do you do that? You can't get rid of the water source because people need it for survival as well. So it's not just an easy cleanup. Like on some island nations, I think they've done this on Costa Rica and some neighboring islands. We watched this video not too long ago of an older woman, and she had a bunch of flowers in a garden all over her house, all around, and they were all in buckets of water. And her house was a breeding ground for mosquitoes. I think it was on Zika that we were watching it. And they had this team that came in, and like their job was to clean up all the water sources. And they taught her why this is bad. But it's like you said, Smike, it's a ton of money and a ton of leg power and maintenance as well. It's not a one-off thing that you can do. But what did they do in the paper? And this might be a bright side for you, Dame Karen. What they did was they took an existing insecticide that has been approved for use, and that is in theory not harmful to humans. Now, everything's harmful to someone in a certain dose, right? Anything beneficial, like a dietary supplement that's good for you, or like something positive at a certain dose, is a toxin. And that's just like that was our intro to toxicology, was everything is a toxin at certain levels. So take that with a grain of salt. But what they did in this video was they wanted to get a crystal structure of this drug. And the way to do that is to heat it up. And that gives you a crystalline structure and appearance of what exactly is happening on the physical level of this solid drug that you're using to kill the mosquitoes. And so they put it in the microwave, and what they found is the structure of the crystals changed. And they were like, well, this isn't really what we wanted to do. We wanted to try to analyze a little bit more of the fundamental properties of the crystal, but let's see what happens. And what was cool was they find that this changed crystalline structure made the drug far more efficient at killing off mosquitoes. And it's something that they did in a microwave that we have in our kitchen. They did make the point of you should not use this microwave for food now.
Now, is that because cannoli's on top of it right now, or is that
it worked?
Yeah.
Oh, that's a great question.
Yeah. I don't know if you can reproduce that without cannoli.
I don't know. Well, I don't know if they had a cannoli, though.
Okay.
Or if they had the cannoli. I guess I don't know. Is it a cannoli or the cannoli? Our cannoli cat or another kid? I don't know. Don't know it's remote. It can list hundreds of cases and signs good intentions and result in bigger problems. Exactly, Kennedy. Grain of salt might be above my allowable sodium allowance. We need hydrochloric acid for blood and associated evolutionary adaptations so our bodies are immune to it. That is a lot, Spectre Flight, to ask for, I believe. Canola is singular. It is, Kennedy. You're absolutely right on that. Microwave is barley spot. It is, but Smikes, Andrew's right. Like the past couple of days have been cannoli on top. See that it's toxic to aquatic life, particularly fish. Generally considered safe to use around humans. It is a neurotoxin. So astronomy showed the way that this is being used before they did this to the drug, it was being sprayed on nets that were going to go over your bed while you slept at night. So it was a protective measure against the mosquitoes actually biting you versus spraying it. So it was not being aerosolized and distributed to the animal population. And then, in theory, if the nets are if they're disposed correctly, which is a big if, they should not be getting into the water supply. So, but given that the crystal structure has changed and made it
in dreams, one is not tethered by earthly limitations.
What does that mean?
Come.
Sleep well, Big Gamey. Sleep well, you handsome devil. Thank you for hanging with us, Big Gamey. Sleep well. What's the difference between DEET and DDT? They are two different chemicals, Leia. They are two different types of insecticides. DEET is more modern. DDT is pertinent during the 50s, I believe. So it's two different chemicals. But yeah, so because they put it in a microwave, they found it all of a sudden became more efficient. And I believe it's in the paper. There's a picture of the two different components. They put it in the supplement. Well, here is the image. And they did a regular light microscope, like the one that we have here. You just did it's a phase contrast microscopy. You just shined a light on it and looked at how the properties change. And you can visibly see the reflection patterns even of these. Like they look almost like salt crystals that you look at under the scope. They look very different from the heat-treated and the untreated version. So on the right-hand side is the heated one. On the left-hand side is the unheated one. And the heated side very well killed mosquitoes that were resistant to it beforehand. So they were resistant to the unheated version. And they killed mosquitoes that were not resistant to it either. So it didn't have any... It was like a brand new drug on the market, or a brand new insecticide. Now, why this is exciting is because I wondered if it would also work on, and they hypothesized in the paper, on other drugs. So we know we have a bunch of resistant bacteria against certain certain drugs that we create. What if we went the same route of just rearranging the crystal structure in this way? Could we also bring back drugs that were thought to be at a loss because there was so much antibiotic resistance built up against it? They didn't test that out, but the fact that it worked on insecticides is a really big deal, and it didn't just help a little bit, it was a huge help against these. Release millions mosquitoes or malaria adapts and makes use of CRISPR tech near impossible. So Kennedy, that's the potential something that happened there. Yeah, potentially. And so, yeah, this is... I thought it was a really cool story, and there's lots of different potential implications that they have in this paper, which I thought was really neat. And I thought, let's jump into a little bit about why using insecticides, and then we'll jump into the next topic for the night. Let's see, where is this next video? Well, we have a generic pesticide video, which also includes insecticides, that one. But yeah, y'all, it's a microwave. Like, people everywhere have microwaves. Like, that's crazy. Again, the interview is like they didn't even go out to do this. They were not trying to do any kind of increase in efficacy of this insecticide. They were just like, I'm trying to look at the crystal structure. It's a biochem lab. And all of a sudden they're like, oh, we may have found something that could be really helpful. And the thing that gets me is already the animals that were already resistant lost all that resistance, went up against this exact same drug as just slightly rearranged. Like, you didn't switch out the recipe, you just changed their structure by putting it in a microwave.
Yeah. Apparently.
It's wild to me. Yeah.
In 1845, Ireland's vast potato fields were struck by an invasive fungal disease that rapidly infested this staple crop. The effect was devastating. 1 million people died of famine, and over 1 million more were forced to leave Ireland. Nowadays, we avoid such agricultural catastrophes with the help of pesticides. Those are a range of man-made chemicals that control insects, unwanted weeds, funguses, rodents, and bacteria that may threaten our food supply. They've become an essential part of our food system. As populations have grown, monoculture, single-crop farming, has helped us feed people efficiently. But it's also left our food vulnerable to extensive attack by pests. In turn, we've become more dependent on pesticides. Today, we annually shower over 5 billion pounds of pesticides across the earth to control these unwanted visitors. The battle against...
And so these are including insecticides as well, and it presumably include, like, the same metric can also be included on ones that we're trying to keep away from us being eaten as well, like the mosquitoes. And yeah, exactly, Kennedy, is because, so the mono food means that they're genetically no longer diverse, like bananas are clonal, and so if a disease breaks out, it's going to wipe out all the bananas on the planet. The same thing with corn, which is why you constantly have to make new pesticides as well. And that's also a question of, does this study, though they looked at one that killed mosquitoes, I wonder if there was some kind of pesticide, like against a caterpillar or something that ate your bananas or your corn. If you microwave that pesticide, I know it sounds so goofy, y'all, but if you microwaved it and you changed the crystal structure, if all of a sudden it became much more efficient again.
Although I would not recommend trying to microwave whatever pesticides you have at home.
No, that is a disclaimer here. Do not microwave your pesticides. You're, like, I'm sure they had like an air vent and like very like in a very controlled setting, right? But it's just interesting to think about, like, if it would be any better or not.
Yeah, that is weird.
Also, risk that pesticides will kill humans. It depends, Leia. Usually these, when are released, there is testing on mammals and to ensure that it's at a concentration where if you were not to have washed your piece of corn and you ingested some pesticide, you're not going to die. Because that would be... people will be dying left, right, and center, right? So that's not an issue. But however, it is important to point out that if you're increasing the efficacy of these insecticides or pesticides with the microwave and changing the crystal structure, that might change the dose that you would want to release it at. Because it might be that it's so much better at killing these things that it might, the threshold to hurt you might have been lowered. Maybe. Long-term cancer. That's so astronomy show. That's the thing. Like, you have no idea what some of these long-term effects are, right? Because on a scale of study, you just don't have the time. Like, we are racing against time to release these chemicals to control populations of insects, of bacteria, of fungus, blah 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 blah. And so there's no, like, what are you going to do with it, right? And so the long-term studies you're not going to be able to have time for. And yeah, now some of these are carcinogenic, so that means they are cancer-inducing. And honestly, I wouldn't be surprised if most of them are, but just at such a high concentration that you're most likely not going to encounter it. But because most everything can cause cancer at a high enough concentration, which is, but the studies that say that, like, if you have Splenda you'll get cancer, while true, the amount of Splenda that you'd have to ingest is like your own body weight every day for years before it'll hit, right? So it's not actually, you're never going to be in a physiological position to be put in, but then it's sensationalized by people of like, I'm going to get cancer from Splenda. I have Splenda every day, and I don't yet have cancer. Exactly, Kennedy. That amount of sugar would be exactly, you die from that anyway before you get cancer. Testing on animals doesn't have the same effect as if you test on humans. That is not true, Leia. So animal testing is called it's a model organism, so that you can model what happens in a human on a closely related animal. That is the only way that we do drug studies, to try to figure out if it can help someone. And yes, it's a model, and no, it's not the most perfect way of demonstrating what happens in the human. It is very predictive, and this wouldn't be done otherwise if there wasn't a prediction method of it.
At what part of the process then would mammalian studies be, from like conception to prescription?
Depends on a lot of factors. It depends on what the drug is targeting. Do you know what it's targeting? Because sometimes you don't know. Sometimes you start off with doing a drug screen. So you can buy a kit of 4,000 molecules, let's say. And then you want to do a high throughput, fast assay to figure out what it works on. That means you're either measuring a behavior or something in cells. If it's in cells, you have a 96 by 96 well plate with cells, and the night Jern, sleep well Jern, and hello iocane powder. You put in the drop of drug at different concentrations with a couple of replicates and see what happens. And usually it's measured with light. So you put the dish into a machine, and it gives you out a light measure of these little tubes individually. And then you're looking for a decrease or increase in light depending on how you adjust your experiment. And then as soon as you see changes, you're like, okay, let's hone in on this drug. Replicate it again. If it happens again, then you can change, like, let's modify this drug a little bit. Because and then as soon as you modify it, it becomes proprietary and you own it and you can patent it.
That's where the money is.
It is. I mean, but because the process gets really expensive.
Yes.
So there has.
Well, you're doing this over and over and over, and that's step one.
Yeah, that's step one. Step two, let's say we have unlimited money. Let's throw it into a fruit fly and let's see what happens. And there, let's say we're trying to. Actually, I did this. It was a neuropeptide F inhibitor. A neuropeptide F is in the brain of the insect, and the neurons talk to the ovary. And the idea is if you lower NPF levels in the brain, it actually pops the ovary, like the cells start popping. So you start feeding the animals different. You manufacture the drug that costs a lot of money because you want a pure form. You have to figure out the dose of it. You have to figure out like a dose response curve, how long you have to feed them to get a certain amount of an effect. What the effect is you're measuring. Replicate. And then if you wanted to test its safety on mammals, you'd probably go into mouse next. And then, and also human cell lines, as like, let's say skin cells, because that's the most likely what it's going to encounter. And then if it's anything similar to human drug testing, you do high dose drug tests on healthy individual humans. And then you test like low end on sickly humans to get like a curve of like what in a very controlled setting. And so we're talking years now.
Yes.
Right.
Yes. Yeah. And what were you talking about? That $200,000, grant.
You can't do it.
That's all of this is infeasible.
It's why usually what happens is as soon as you discover a drug in the lab, and let's say we do the fruit fly step, you sell it to a drug company. And drug companies will have a bunch of these that they're pulling along, and maybe there's 10 active projects and one of them pans out. So the reason why you might, and I don't like that drugs are so expensive, but the one reason why they're so expensive is because that one drug that made it is funding the 9 other failed attempts. Yep. And a lot of them, like, they get through so many steps, and they get to phase 1 or 2 clinical trials in humans, and then everything falls apart because of safety concerns, right? And so they are heavily tested. I don't know for this drug that we talked about with the microwaves of different, like, changing the efficacy of it, if that would require additional testing or not. Mm-hmm. So that would be one important thing.
Well, and so I actually have a friend who works at the FDA that audits and goes through those types of processes, you know, susses them out, looks at what they're doing, seeing if they're appropriate or not, and you know, presents findings and stuff like that. And I hear that all the time, you know, look at you know, this bed study or that bed study, or I don't know why they were using this measure, and this minutia that it comes down to, right? Because you have such stringent ways you're supposed to be looking at the test findings, right? You try to fudge that left or right, like I can see that from a side that's trying to push this through because that's what it would be funding, those other, you know, failed experiments. So you have a lot of checks and balances in the system.
Yeah.
Way before you ever get to anything being widespread.
And our friend Iacane Powder here is actually a clinician, and that's what they do. So Iacane, did we get it? Did we get it right on the explanation?
Especially insects has marked agriculture's long history. Records from thousands of years ago suggest that humans actively burned some of their crops after harvest to rid them of pests. There's even evidence from ancient times that we recruited other insects to help. Later, as large-scale farming spread, we began sprinkling arsenic, lead.
There's genetic tools that we're trying to kill these pathogenic-carrying mosquitoes. But on top of that, there's also insecticides. But we're running out of insecticides because there's all this resistance being bred into the mosquitoes. And the question is pretty simple. How do we combat that? And so what the researchers did was they put the insecticides in a microwave because they were trying to look at its crystal structure. They wanted to analyze how the crystals change under different stressors. And so what you can see is this image on the left is the regular drug. It's a solid state of the insecticide, and then here it is microwave. You can see it looks physically different. Chemically, it's the same. It's just the crystals look a little bit different. Thank you, Tatum, for the lurk, you amazing soul. They just look physically a little bit different. And it turns out their chemical properties are also now different. So animals that were resistant to the drug all of a sudden are very susceptible to it. Again, chemically has not changed. The only thing that's changed is the structure of the chemical. And that has huge implications for not just insecticide on this front, but other insecticides that we've deemed no longer useful because the animals have been bred resistance to. On top of that as well, the implications of it could be for drugs that are involved in human importances as well. For example, resistant bacteria to certain drugs. If we can do the same thing to them, imagine the potential of what we'd have. How does the chemical unalive the bug? It's a neurotoxin, Boyds. So it shuts down the neurons, and then it's kind of grotesque. The bug just dies because it can't eat, it can't drink, and yeah, that's how it ends up dying. A microwave crystal, says Leia, look like CD laptop video player stuff. I can see that, Leia. It feels a little bit like that. Worried about my boiled mail now. It might have crickets and crickety kisses on it. Anchored, listen, if you're blessed, it will have extra crickets and kisses on there. In fact, Anchored, just for you, he's going to kiss both sides of the coin. Yeah, with lipstick. It'll even be marked. It'll be born into the wood, and you'll try to wash it away, Anchored, and it won't leave. It won't leave at all. Guys, go follow Anchored Albatross for all things, like, to help her on this journey of safety, emotional safety. She's also an amazing wood burner extraordinaire. Anchored, Andrew saw your wood burning piece today, the Winnie the Pooh map, and
I took a picture of it immediately. It was hanging right up in the baby's room. I was like, I thought it looked good, like on stream here. It just blew me away in person. That was amazing.
Anchored, what do
So
you
cool.
what do you use for the color? I know as you painted it in, but was it watercolors? I know we were talking about that of how you added in the extra detail of that, because that was, I want to make sure that you were doing that. Yeah. The logo she made for StreamScribe.
Yes, absolutely. Yeah, it's gorgeous.
Watercolor pencils and regular colored pencils for that one.
Yeah.
Anchored.
Yeah.
Everyone who sees it is floored.
Yeah. Oh, it's you don't see that anywhere, And it's like, I didn't know that was possible, and or to look so beautiful. Like it's stunning.
And the burnt rims too. It's just like totally. Yeah, it's stunning. Tell me all the secrets, Anchored. Would you still eat the crickets if show them to us under the microscope? Tandem, look, I will show cricket legs under the microscope if Boyd sends them to me. Absolutely. I don't think it could gross me out anymore, to be honest, with those legs. Boys, that is like the crickets. When you're eating them, they do get caught between the teeth, and it's just kind of grody, and you're still wiggling for hours. And it's a whole thing. But yeah, so trying to kill insects, this has huge reaching implications. And we were also, as y'all rated in, we were talking about the benefits of insecticide and just a broad level of what all things that we could be used for. And again, we were talking about how there is overuse sometimes that could lead to resistance to these, and maybe we'd be able to get rid of that resistance by simply doing the simple microwave test. Again, that's a long stretch. It might not work for everything, but the fact that it works for something is a pretty big deal. So it's a really exciting study. Anchors says we're 21 follows away from 1,000. Anchored, ma'am, what are we doing when we hit 1,000? Another 24-hour stream? I'm excited, Anchored Albatross. Anchored, we got you. We're going to help with that poosh. We're going to help with the poosh, Anchored Albatross, as we always do. And the 1,000, ma'am, hitting that 1,000 mark is such a weight off the shoulders. It gets, I'm not saying that you will never look at those numbers again, but it just feels so much better when you finally have that piece.
Where's my drink?
Thank you.
What drink?
Yeah, thanks for
a little parched. Chilling and watching that number grow so many amazing humans, just like Anchored Albatross. Lovely Derek Devon of Moco Made. Please go follow the amazing Moco Locos. We love you, Derek Devon. Love y'all. Okay, so Boyd says, how do the bugs get resistance? Natural selection of those who survive and pass on their genetics, or maybe not natural selection, but those who survive. So, Boyd, I love how you've asked the question. Because you're right, it's I guess it might kind of be natural selection, but on an unnatural scale, because naturally you're not going to have insecticides or pesticides sprayed, right? Like, that's not a natural threat. But it's natural selection because the ones that are killed off are now no longer in the gene pool, and the ones that survive have been selected for naturally in terms of life or death. They're still alive, and usually it's a very small subset of the population that lives. And that's, boy, it's not just for insects, but the same principle applies for when you have antibiotic resistance bacteria or fungi or something. There's always a small population that might have some, usually it's a mutation at that stage, that isn't beneficial for the population, so it's only in a small percentage of individuals, because there's no reason for everyone to have it. There's no selection pressure or advantage to have that genetic quirk at the time. This is also why, boys, when you do drug tests and you do infection, like these infection curves, you want to make sure that you use multiple drugs, because the likelihood of there being multiple drugs that will negatively, like, that will kill off the animals is much higher likelihood that you'll kill off everyone. Because the likelihood that one thing has multiple resistances at that stage when there was no selection pressure on them before is much lower than having a image here, Saul. That yellow is actually all muscle tissue. So all of that muscle tissue is what they're using to hold up the leaf. And so without those muscle, so for those, that's the bite force. So you can see here what all this yellow is connected to is this bottom base of the jaw. And that bottom base of the jaw is what allows these animals to either bite you or also allows them to lift the leaves up. And this is the same not just for leafcutter ants, but for other ant species as well. So there is a fair bit of muscle there, but for walking and movement, it's all on a hydraulic system. There is some of this muscle in there as well, but again, it's much thinner. It's a support system rather than the primary system for it. And then Saul, if you want, I can dig up the image I was talking about. So what I did was you can dissect away the cuticle or the outside exoskeleton, and you can put in a stain that marks muscle tissue inside the head, and it actually fluoresces. It'll glow a certain color, and it'll actually tell you everything that's muscle from brain and other tissue. Actually, it's neat as the brain is actually way back here, and it's super tiny in relative to the rest of the head because they're not designed. These are designed for carrying things and for attacking and not for thinking necessarily. So it's like proportionately a different part of their head. I can show you like what the... And then there's the undissected version of the head there as well. How does muscle function without blood supply? So they do have blood, Saul. They just don't have veins, arteries, or capillaries. They have free sloshing insect blood or hemolymph. It is just free flowing. So it's constantly bathing everything inside their bodies. They do have a proto-heart. The blood doesn't rush through the heart. Instead, it's just a muscle and it contracts inside their body. And that causes there to be enough pressure to move the blood throughout the body. They also do have a blood-brain barrier similar to us, and small molecules can cross in between the blood-brain barrier. There's a lot of similarities with higher order mammals and organisms, but there are some key differences, in particular that open circulatory system. Crabs have that as well, Saul. It's like when they have an immune response, it actually barricades off the pathogen like in a different part of the body to make sure that it doesn't slosh around to the rest. So it's a different way of having your immune response as well. Yeah, but great question. All right, so for the Raiders coming on in, we were talking about insecticide resistance and how you can actually regenerate an existing insecticide using microwaves. And there again is the Discord link if anyone's interested in joining the Discord as well. Science continues on there. Free-flowing blood is used for hydraulics. Yes, because it's all the liquid in the body. So that's all, and also why when they dry out, they shrink right back down on that front. Heard about the blood-brain barrier a lot, not sure how it exactly works, especially relates to alcohol. So certain small molecules, Leia, can cross the blood-brain barrier. No, Saul, don't you dare. Don't you dare, Saul. This whole channel is around questions. I might have something planned for the stream, but I promise you we're not getting to it. We're going to be getting one topic tonight. Okay, Debs wanted 2. That's her prediction. Gwen predicted 10, as she always does. But there are some streams, Saul, that we do science news and we hit 3 topics or 4, and there are others like tonight. We went in depth on why malaria, like what about malaria, and then like how it's transmitted, how you can use genetics to counter malaria, and then how you can also utilize insecticides to fight them off. So there's a lot of avenues that we're going on. And so we thoroughly hit up the topic, and there were just questions, and that if there's questions, that's all the better it means because people are interested.
Yep.
If I were here, I would request a special pool story. Torres, we didn't do a pool story in person. I know.
What a miss.
It is.
We'll have to hang out again.
Yeah, that's the solution towards night. Maybe tomorrow. Yeah, are you coming for the barbecue towards night? Coming for the Professor night? So Leia, small molecules can cross the blood-brain barrier. The reason it exists is that large pathogens aren't going to cross into your brain like worms and like if you get a tapeworm, it's not going to use your bloodstream to get to your brain and eat out the brain. So it is a protective measure, but there are small molecules that can cross over, such as alcohols, and especially in certain breakdown and metabolized states that can go through the brain. Now, sleep well. Are there lungs? So they don't have lungs. Instead, they have tubes called sphericals. They go from the exoskeleton, and the tube just goes into the body and stops. It doesn't go in and connect anywhere. It's just, imagine if you popped open this animal, there would be a bunch of tubes running in and out, and it's through diffusion. So it's high concentration of air diffuses to low concentration of air that's in the body, and that's how they breathe. That also means there's a limiting factor in how big they can get. Because diffusion only works on smaller scales, the amount of oxygen that we have in the environment isn't enough to allow the animals to grow giant. Versus if they had lungs, they can get bigger. Does that make sense, Saul? What are we having, Draft Killer? So Draft Killer, I have ribs. We have pork loin that I'm going to marinate overnight. We have ribeye. We have beef tenderloins. Beef tenderloins and pork loin. Hamburgers, hot dogs, and imported Hungarian sausage that I have been saving for a special occasion. Like, it's been frozen away for this special occasion. Yeah. And then we're also going to have vegetables like corn and carrots and peppers, and Lita will have her fake burger and like all that jazz. Yeah. But the rest of it is hopefully be delicious. Gwen, come on, Gwen. Chicken and pork rotisserie style on split. Yes, Leia, that is delicious. So an ant can basically hold its breath. So Saul, the air doesn't go in and out of like an insect's mouth. If they breathe through their skin. And actually, Saul, when you mark an ant in a colony, like let's say you want to track an individual, most common way to do it is to paint it. So you put a paint spot on its abdomen. And actually, if it gets covered in the paint, it suffocates and dies because they breathe through their skin. And so it's weird. It's like not through their mouth, right? Because that's our standard. I don't know. Maybe it's weird only to me. That's always what I thought of. Like, I see a mouth, I assume that you're eating and breathing from it. But instead, it's all through their skin and their exoskeleton. And it's a gnarly system. It works for them, but again, that's also why you don't see... Like, the biggest one that is, like, Camponotus giga is about the size of your hand, the queen. And there's really not much bigger than that, because that's, like, pushing the extremes of it in terms of sizes. And it's energetically costly as well to be bigger in the insect world. There's a lot of factors at play. But yeah, no lungs. They do have a blood filtering mechanism for toxins, you know, similar to like your kidneys, but not a real kidney. They have ovaries. They have sperm producing glands. They have a gut and digestive tract. If we're talking about poops and peas, as it were, they actually do the same. It's a droplet of, honestly, like filth. It's like just a brown liquid, and that's all it is. It's quite fat. There was a woman by the name of Corey Moreau. She is a professor at Cornell, and she got some really viral tweet about, because I think it was Cardi B or Beyonce or someone along those lines put out a tweet of do ants even poop? And she responded with the science, and it went totally viral. And that was like the big push, as it were, on like the ant biology front, where people had never thought of it, and it was just really cool to be able to get out of it. How is it proven that ants can't have complex thoughts? Because the push didn't even register. That was second. Didn't even register. It hasn't, Saul. It's assumed that an ant cannot think as complexly as a mammal. So there's regulations in the scientific field that if you're working with mice, you can't do X, Y, and Z to them because it's animal abuse, right? And everything that you do to them has to be justified and done in a certain protocol, so they're not feeling pain when you kill them for samples. That's important to do because you don't want the Wild West of people loosey-goosey killing animals. Yeah. But in the insect world, it is the Wild West, where it's like, well, they're not really feeling anything. They do feel stuff. There are pain receptors. They presumably have some level of self-awareness. It might not be the same way as a mammal, but there are presumably some flavor of thoughts. There's an experiment, it's very dubious when it was done, where if you put a mirror in front of an animal, if it recognizes itself, it's said to have a sentience about it. And if you look at these animals, like that study suggested it works. It's questionable because I don't super buy it, in part because of how it was done. it didn't look like it was efficiently performed, the tests, where it's like there wasn't a lot of replicates to it. It didn't seem like it was going to be a very well done piece, which, you know, is unfortunate, but that was my feeling about it. But it's still a possibility, right, that something like that exists. So, to answer your question, Saul, we don't know. It has not been proven. How do ants get enough oxygen underground to get their colonies to be able to breathe? So, Tea Time Cat, that's a great question. Welcome in. Tea Time, there is enough air or oxygen in the air underground for them to be able to osmose that. Like, there again, you're using diffusion. It is going high concentration, low concentration. Inside your body that doesn't have oxygen is inherently low concentration, so it'll directly be going into there. And they don't need that much. Will a cave-in kill them and lose oxygen? Yes. But they, building-wise, are quite spiffy. They will make sounds and call for help. And there's even a scream that they have. They have a scream that they'll do, and it'll bring them on. It'll bring other workers, and they'll unbury the creatures, which I think is really cool. And an insect get cancer? Yes, Saul. Actually, my postdoc work was giving it cancer to insects. Insects to cancer. Cancer to insects, yes. And the reason was when an insect and a human get cancer, it's out of control cell growth. And it was particularly, we're looking at colorectal cancer in insects. And the genetic pathway, the mutation that you can give an insect to give it colorectal cancer is the exact same mutation that a human gets. It's in a gene called APC. And that mutation causes there to be uncontrolled cell growth in the form of polyps in the insect, which is the same thing that you would have in a human. Now, it might look a little different, but you can mark the stem cells in an insect. If you mark the stem cells, they'll fluoresce green. You'll see they start proliferating really quickly, and then they start shooting off more cells. And you just, instead of getting a nice, so the insects don't have this kind of intestine. They have a very flat one. And here, it just starts growing in polyps, and they actually end up clogging their system. And you can target it to particular regions of the gut as well. And so what I was doing, I was feeding a chemical to ants that was turning off that APC gene, and then you looked at the gut, and all of a sudden it was just massive growth there. And so naturally they don't usually live long enough to get cancer, but you can induce it through mutation. Is it always crazy cell growth with cancer cells every time? So Draft Killer, at the most fundamental level, cancer is defined as uncontrolled cell growth. The most basic level, that's what it's an immortalized cell. So it loses the machinery to say, when I should no longer divide because I am genetically compromised. It loses that ability to it's usually a gene called P53, is a very common mutation. It's a master regulator of cell growth and cell division. If that thing is turned off, then that checkpoint to decide if my genome is compromised or not, it goes away. And then the cell just keeps dividing and dividing and dividing. Instant metastasis. Metastasis is you start off Draft Killer with a small population of these cells. Even though they're dividing, it's not, one needs to divide to two, and then those two can divide. And so it's exponential, but they won't move to a different site until they start using up all the resources in a given site. Like if you have lung cancer, right? When they've eaten up and used a lot of the energy there is when they'll they start hitching right in the bloodstream, and they'll land on a different spot, and they colonize essentially like a bacteria a different part of the body, and then you'll have their growth with no the motivation is not conscious but resource motivated. Yeah. All right, chat. Shall we finish up this video? Let's do so. And then that's where we'll be, I think, rounding out the evening. Going back to the thought, can different insects communicate with different insect species? Ooh.
Communicate's hard to define there.
If you define... so Saul, You just waded into a big debate.
Yeah.
The big reason is because the argument is it communication or mimicry? And I actually had in my PhD a paper where it was different fruit fly species communicating with one another about a predator threat. And there we were able to prove it was not mimicry, but rather it was a visual cue transmitted from one species to another about this predator threat. And the reason we could prove that is because the behavior manifested as laying fewer eggs. And what ended up happening was you can genetically block their ability to lay fewer eggs, but they still did this wing movement, and that was the communication. So there it was totally visual. It wasn't a mimicry base, but there's a lot of other ones where it's they demonstrate a mating culture in fruit flies, where if you can teach a female fly to prefer a fruit fly that has this green dust on it, and then like for some reason that's better, and then the fruit fly will prefer the green male all the time, and even their next generation will start. It's not genetic, they like watch each other and they teach each other these things. Is that mimicry or communication? So that's the difficulty, but the idea is yes, they fundamental level they do. Lesebra, no sorries, welcome on in. What species like to communicate with each other the most? Usually being it's not, I wouldn't say this is gonna sound anthropomorphic, a common goal or threat is what they are most likely to have cross-species communication with. Birds are very much like that. Like if you go outside, you'll see a bunch of birds fluttering around. If all of a sudden they disappear because there's a hawk, they eavesdrop and listen to each other's alarm calls, and it's beneficial for you to learn something like that. So usually it has something to do with predation or some kind of threat in terms of communication. I just came here to say my name. May I ask again? Andrew has a degree. Andrew is very specialized draft killer.
Yeah, I do a lot of tech stuff.
Technical wizard draft killer.
Yeah, that's my forte.
Also, moderator extraordinaire. Also, just an all-around amazing person. Like, my daughter's smile is indicative of how good this man is. Like, that, when I told Lita and Lita saw the same thing, she said the exact same thing what I said, which she goes, Ilona recognized a really good person. Mm-hmm. So he's very smart, very talented, but he's also just good people. So, draft killer, if you're not following Andrew, please go drop him a follow, okay? And he gets uncomfortable whenever I do the shout outs, but please go drop him a follow. So I'll ask, do bugs have ears for vocal communication? They vocal in terms of auditory cues, yes. It's not a functional ear. It is a organ called the Johnston's organ, and it is a organ right up here on the shoulder of like a fruit fly, and it's detecting vibrations. So they can detect sound as well. Usually visual and olfactory cues, like smells, are the big things that they're used to detect. And yes, they do. Therefore, they can detect things without like touch without nerve endings. Like these vibrational cues as well. Okay. So let's finish up if we need pesticides or not, and then we'll round out the evening.
It's on crops. But these were incredibly toxic to humans as well. As our demand for more, safer produce increased, so did the need for effective chemicals that could control pests on a grander scale. This ushered in the era of chemical pesticides. In 1948, a Swiss chemist named Paul Hermann Müller was awarded a Nobel Prize for his discovery of dichlorodiphenyl trichloroethane, also known as DDT. This new molecule had unparalleled power to control many insect species until the 1950s, when insects became resistant to it. Worse, the chemical actually drove dramatic declines in bird populations, poisoned water sources, and was eventually found to cause long-term health problems in humans. By 1972, DDT had been banned in the United States, and yet traces still linger in the environment today. Since then, chemists have been searching for alternatives. With each new wave of inventions, they've encountered the same obstacle: rapid species evolution. As pesticides destroy pest populations, they leave behind only the most resistant individuals. They then pass on their pesticide-resisting genes to the next generation. That's led to the rise of superbugs, such as the Colorado potato beetle, which is resistant to over 50 different insecticides. Another downside is that other bugs...
And then, as an aside, it might be resistant to those insecticides, but what if they did the method that was described in this paper, which is microwaving those insecticides, when all of a sudden they become better again? Right, that would be a cool thing to test out.
Get caught in the crossfire. Some of these are helpful predators of plant pests, or vital pollinators, so erasing them from agriculture wipes out their benefits too. Pesticides have improved over time and are currently regulated by strict safety standards, but they still have the potential to pollute soil and water, impact wildlife, and even harm us. So considering all these risks, why do we continue using pesticides? Although they're imperfect, they currently may be our best bet against major agricultural disasters, not to mention mosquito-borne diseases. Today, scientists are on a quest for alternative pest control strategies that balance the demands of food production with environmental concerns. Nature has become a major source of inspiration, from natural plant and fungal chemicals that can repel or attract insects, to recruiting other insects as crop bodyguards. We're also turning to high-tech solutions, like drones. Programmed to fly over crops, these machines can use their sensors and GPS to carry out more targeted sprays that limit a pesticide's wider environmental impact. With a combination of biological understanding, environmental awareness, and improved technologies, we have a better chance of finding a holistic solution to pests. Chemical pesticides may never shake their controversial reputation, but with their help, we can ensure that agricultural catastrophes stay firmly in our past.
That's a good summary on that front. So that's the first paper and the only paper tonight, chat. We did it. We did it. Hi, Dilly the Doe. Guys, go check out Dilly the Doe, legendary streamer extraordinaire. We love Dilly here. Go check out Dilly the Doe. Go check out Dilly the Doe. Let's see, quick question. Is it gonna get weird? Can two different ant species breed like lion and tiger? So, Saul, ant species are really difficult to get to mate the same species in a lab because they have to have a mating flight. Dilly the Doe, thank you for the biddies. That was not required. There's never expected, but always appreciated. Thank you, Dilly the Doe. They have to have a mating flight at certain altitudes, at certain temperatures, at certain densities of population, certain moistures. You cannot mate them in a lab. It's near impossible. Fruit flies, though, you can mate closely related species that normally shouldn't, and you'll produce something like a mule where it'll be sterile offspring. And then you can actually genetically manipulate them, try to make them not sterile. But with ants, is no Gucci. It's going to be very difficult